Hello friends. Today we will discuss about the microbiology of drinking water part 3. Today we will discuss about the coliforms as an indicator and the how to determine the coliforms in a drinking water and it's a process. So let's start. Coliforms organisms, particularly E. coli, are consistently present in the human intestines in a large number. It is estimated that the billions of these organisms are extracted by an average person in a one day. These organisms generally live longer in a water than the industrial pathogens. A healthy person would not normally extract pathogenic organisms, but should an industrial drug infection develop, the pathogens is likely to appear in the feces. Thus, the presence of coliforms in a water is regarded as the warning signals the water is subject to the potentials to dangerous pollution. Classifications of the enterobaxis. Before the part 2 in we already discussed about this enterobacteriaceae group and classifications and the how to different between the lactose fermenters and the non-lactose fermenters. Test to know how to test for the coliforms present in a water. So different types of tests it's present. It first it's indole, second it's methyl red and the Voges-Prosakaur. Third one is the citrate utilizing. Fourth it's the H2S production in the enzyme, urea hydrolysis, motility, lactose fermenters, glucose fermenters and the gas productions, decarboxylations of amino acids, fermentations of sugars, reactions on the selective media. First, IMVIC test. So what is the IMVIC test and what is the full form of the IMVIC? IMVIC is I. I means indole, M means methyl red, V means Voges-Prosakaur and the C was the citric test. So this is the IMVIC test. The following four step test comprise as a series of important determinations that are, are collectively called the IMVIC series of the reactions. This IMVIC series of reactions allow for the differentiation of the virus numbers of enterobacteriaceae. IMVIC indole test. In the IMVIC test, first test, it's discussed about the indole test. What is the principles? So principle it's a certain microorganisms can metabolize tryptophan by the tryptophanase. Tryptophan it's a protein and it's amino acids and the tryptophanase it's a enzyme for degradations of the tryptophan. The enzymatic degradation leads to formations of the pyruvic acid, indole and the ammonia. The process of indole is detected by additions of the CoVac reagent. You can see this in the chart. Tryptophan is its amino acids, tryptophan its amino acid and the tryptophan its enzyme for the degradations of the tryptophan, and after the degradations of tryptophan it's formed in indole, pyruvic acid and the ammonia, and the after the additions of the CoVac reagent and red color appear in a organic layer. Results. A bright pink color in the top layer indicates the presence of indole. When present in the medium and add to the CoVac reagents, you can see this the pink color ring, and the absence of color means that indole was not produced. It's indole negative test. Usually indole positive test seen in E. coli and the enterobacters it's a give it a negative test. Second test it's a methyl red and Voges-Prosakaur test. You can see this in the chart. The glucose and the glucose fermented by the two two pathway. First pathway it's a acidic pathway, and the second pathway it's a neutral pathway. Acidic pathway considered as a methyl red test, and mixed acidic pathway mix acid, and when the medium mix acid, pH is less than 4.4. Methyl red indicators methyl red it's indicator add into the medium and form its red color, stable red color. Remember this, in the stable red color, so the test is a positive, and E. coli it's give it a positive test. Now we can discuss in a Voges-Prosakaur test. It's a neutral pathway. Add to the acetyl methyl carbenols solutions, and its solution in a pink color, and the Klebsiella and the Enterobacter give its a positive test for the Voges-Prosakaur. Butadiene glycol pathway of the glucose fermentation. In the fermentations, in the butadiene glycol pathway, pyruvic acid its acetone in butylene glycol, acetone and the butylene glycol are detected by the oxidations to diacetyl at an alkali pH, and the additions of the alpha naphthalene, which form a red color complex with the diacetyl. Important biochemical property used for the identifications of the Klebsiella, Enterobacter, Serratia. Voges-Prosakaur reactions. What is the reactions? So you can see this, the acetone and the butanediol glycol are detected by the oxidations of the diacetyl and the alkaline pH, and the additions of the alpha naphthol, which form the red color complex with the diacetyl. Usually Enterobacter can responsible for the alkaline pH, so the Enterobacter it's give it a positive test. The productions of essentials the butylene glycol by the glucose fermentation, it's an important biochemical properties used for the identifications of Klebsiella, Enterobacter and the Serratia. MRVP test, it's a form of methyl red Voges-Prosakaur test method. Inoculated the test organism into the MR and the PP broth, incubated the tubes at the 37 Celsius for 24 hours. For methyl red, add to 6 to 8 drops for the methyl red reagents, and you can see this stable red color. And for the Voges-Prosakaur, add to the 12 drops of the alpha naphthol and 4 drops of the solutions of the KOH 40% KOH solution, and the mix well, and you can see this pink color. This is results of the MR test. You can see this negative test in a yellow broth and the positive test in a stable red color. It's positive test responsible for the E. coli, and in the Voges-Prosakaur test, pink color, it's a positive test for the Klebsiella and the E. coli. And but citrate utilization test. Citrate utilization test, it's so important for us, because the citrate it's a source of carbon, and in this medium we cannot add to the any type of other carbon source. It's only and only carbon it's present in a citrate. So principle, citrate in a form in a pyruvate, and the pyruvate in a CO2 and the sodium and the H2A. So sodium and the carbon dioxide join it, and the form it's a sodium bicarbonate, and sodium bicarbonate it's a basic compound, it's a alkaline, so pH is increased, and use it's a Siemens citrate medium, alkaline pH, and the add to the bromothymol blue, it's a indicator, it's a dye, add to in this medium, and blue color blue color seen. The positive test. The positive test, it's Klebsiella, Enterobacter and the Citrobacter, because these three organisms are produced for the alkaline pH. And the negative test in E. coli, because E. coli is the productions of the acid and gas. Citrate utilization test method. Streak a Simmons citrate agar slant with the organisms incubated at 37 Celsius for 24 hours. Citrate utilization test, you can see this result. The positive test is the Klebsiella and the Enterobacter, green to blue, and the negative test it's E. coli. Examination for growth. Growth on the medium, it's accompanied by a rise in a pH to change the medium for the initial green color into the deep blue. It's an IMVIC test. A series of 4 tests consists of the indole, break down the amino acid tryptophan, methyl glucose oxidations, Voges-Prosakaur, productions of the neutral and the products, and the citrate fermentations used to differentiate the Enterobacteriaceae group, and we will look at each test individually. You can see this. The first is the indole test. Medium it's SIM agar, and the substrate it's a tryptophan. You can see these tryptophan structures, and the tryptophan is enzyme degraded to tryptophan. It's indole pyruvic acid, and the indole present in a broth add to the coax reagent, and you can see this the ring form to junction. Methyl red. Medium it's a MRVP broth. Substrate it's a glucose, and it's including the GPP broth, glucose plus H2O. In the E. coli it's produce the acid, lactic acid, acetic acid, and the formic acid. These three acid formed by the E. coli, and the produce of gas it's CO2, and the pH it's low 4 point. And the Enterobacter it's acid. It's formed two acid, lactic acid and the acetic acid. Lactic acid form it's ethanol, and the acetic acid it's acetyl methyl carbonyl, and the H2O and the form of the gas, and its pH it's 6.5. And the methyl red, you can see this low pH in a unstable red color, but in a high pH you can't see this red color. So E. coli it's give a positive taste in a methyl red test. Voges-Prosakaur test. Medium as usual GPP broth, and substrate it's a glucose and the oxygens. Acetic acid form the fermentations, and 2,3 by butanediol acetyl carbonyl plus H2O plus CO2, and the pH it's a 6 point. And the Voges-Prosakaur test it's a alkaline medium, and the Enterobacter it's a give a positive taste. And the citrate test. Medium Simmons citrate agar substrate is a citrate, and citrate utilizing enzyme only to utilize the citrus, and the requirement for the carbon it's fulfilled, and the give the positive test is green into the blue, and the Enterobacters are give it's a positive test. Thank you.